Thank you all for joining us. This is our penultimate bite-sized research webinar, so thank you for taking the time out to join us today. It seems like yesterday that we started the series, zoomed through them. But if this is the first time that you joined us, we would ask that everybody stays on mute, please. You are already on mute as default, if you can still remain that way. If you have any questions throughout the webinar, we ask that you use the chat function. If you have two screens or you're able to open the chat as well as look at the video, we'd recommend that you open the chat so you can add any questions as they spring to mind. There are no silly questions. If you think of something, pop it into the chat. I'm joined by my colleague Vicky, who will be monitoring the questions that you're asking, and she will put them to our speakers at the end. So one last thing, just to remind you that this webinar is research-based. It isn't about personal treatments or circumstances. If you do have any questions along those lines, our support and information team are available, and you can email them at support@bcrt.org.uk. Okay, I'm going to now hand over to my colleague, Dr. Zoe Davison, who is our Head of Research, Information and Support, to introduce this week's speakers. Zoe?
Hi everyone, and welcome to the penultimate webinar. So this webinar is focused on giving you some progress on our Chordoma research program. So last year, we actually managed to fund 2 projects focused on Chordoma, and this is going to be looking at the progress made on one of those projects. And these projects are building on some of the work that we funded before and work that's been funded by other charities and organisations. So along with the projects that we've funded, our sample collection grants have allowed around 600 Chordoma samples to be collected, which is really fantastic. So they can be used in research projects funded by BCRT and other organisations, and hopefully they'll make a real difference to the research that's going on. So it gives me great pleasure to introduce our 2 speakers. It's a very collaborative project. So Dr. Lucia Cottone and Dr. Adam Cribbs, over to you.
Thank you. Let me share the screen. Okay. Can you see my screen and hear me? Great. Thank you, Zoe. Thank you very much for inviting us to give this talk today, which is about our progress in Chordoma research. I'm Lucia Cottone, and Adam is going to talk through the second part of the presentation. So we really would like to tell you what we have been doing, also thanks to the funding from BCRT. And the project that we have been working on is really a collaborative project, collaborative work between 2 groups that have quite diverse expertise. On one side, Adam and Professor Udo Oppermann at the University of Oxford. They are experts in chemical biology, epigenetics, and bioinformatics. On the other side, myself in the group of Professor Adrian Flanagan at the UCL Cancer Institute, we have tried to contribute our expertise on chordoma biology and pathogenesis. So the focus of this project is chordoma. Chordoma is a primary malignant bone tumor that can arise anywhere along the spine from the top, from the head, through the coccyx. It's rare, so 1 in a million is the incidence of this disease. If we talk through numbers, as Zoe just mentioned, about 40-50 new cases of chordoma are diagnosed every year in the UK. So no 600 that she just mentioned, actually it's a very important number for the chordoma community in terms of research that can be done. It is a slow-growing tumor, but it's also very aggressive, and especially if it occurs in areas that are close to delicate organs such as the brainstem. Even if this tumor is rare, the community has made significant progress over the last 10 to 15 years, and the research really has improved. This is a picture of a chordoma sliced and looked through the microscope, a histological picture. And you can see that it's characterized by these big cells here, these big vacuolated cells, which are quite peculiar of the histology of this disease. However, for a pathologist as well, diagnosis of this tumor is not always so straightforward. So one of the quite important breakthrough discoveries that has been made a few years ago by the group of Professor Flanagan is the identification of a molecule which is called Brachyury. This is again a chordoma, but have been stained to reveal the presence of Brachyury in the cancer cells. And the brown staining here is identify the nuclei of the cancer cells in which Brachyury is present at very high levels. When Brachyury was identified, and effectively every single chordoma sample expresses Brachyury at high levels, and this is very unique. It's probably one of the few tumors can be used to identify this tumor in a very precise and unique way for diagnosis. But when it was identified, it was really clear that it wasn't just a bystander, but it must have been involved in the pathogenesis of the disease. Indeed, we know that Brachyury is involved in the development of the spine, in particularly is important for the development of the spine in the embryo. And we know this because there are some animals, dogs, mice, and also some cats that have alterations in the gene that codes for Brachyury. And these animals display quite important defects, such as they don't have a tail. And this is quite a striking example of this Manx cat. And indeed, the name Brachyury derives from the Greek name, brachus, short, and ura, tail. So short tail, so no tail if Brachyury is altered during normal development. Not only it is involved in the pathogenesis, but actually it really is fundamental for the tumor to grow. So we knew this because some experiments were done a few years ago. Chordoma cells were taken and grown in the lab, and these chordoma cells were genetically modified to delete the expression of the presence of Brachyury. And it was very clear, very striking that without Brachyury, the growth of the cells was completely hampered. So if you look at this graph where you have the control curve of normal chordoma cells that can grow quite rapidly in the lab over time, without Brachyury, their growth was completely blocked. So this revealed really that this molecule, which is there, which can help us in the diagnosis of the tumor, can also be one of the, or the Achilles heel for this disease. And really, by hitting this Achilles heel, we could have a chance. This has been demonstrated in a Flanagan lab, but also many other groups have repeated these experiments and validated these findings. The identification of such an important Achilles heel for the disease, it's very important, especially because unfortunately few treatment options are available even now for chordoma. Chemotherapy is not used because it's not effective. Surgery is the most important treatment, but it can be extremely challenging, especially for those tumors which are hidden in delicate parts of the body. Quite exciting results came through a few years ago when novel drugs were identified, and in particular the EGFR inhibitors identified by our groups and others in the world are now being tested in clinical trials, and we will hopefully see the results soon. However, we know that there is no one magic bullet for every situation. Not all patients will respond, recurrences will occur. So we really need to identify more targets, and still we need to develop more drugs. Moreover, importantly, even if we know that Brachyury can be the Achilles heel, and it is indeed an ideal target that could completely wipe out the presence of the cancer cells. However, this molecule belongs to the class of transcription factors, and transcription factors are very well known to be quite difficult, if not impossible, to be targeted pharmacologically by compounds that hit these transcription factors directly. So it is very difficult to come up with a drug that inhibits brachyury directly. And in collaboration with Udo, Adam, and Adrian, when we were thinking about this problem, we thought, can we follow another approach? And can we circumvent the issue by targeting brachyury indirectly, by shutting it down in another way, not by blocking it directly? And we came up with the hypothesis because we know that there are some epigenetic drugs that are available, and because we knew our epigenetic research so far. So we're talking about epigenetic now, and when we talk about epigenetic, we refer to all the biological mechanisms that occur so on top of the DNA structure in the cells. So the DNA is present in the nucleus, as you can see here in this picture, but it is not just present as a free to flow molecule. It is organized in a 3D structure, which is highly organized, and the compaction, the organization of the DNA in this 3D hierarchy is possible because there are some molecules which are called histones that physically wrap around the DNA, creating these chains of the DNA and making it very compact in the nucleus. Now, these histones have a scaffolding function, but also they harbor some modifications. These are just chemical groups that are attached to the histones and can decide and modulate in a very, very fine way whether the DNA is more compact or is more loose in the nucleus. So epigenetics it's actually a very important biological mechanism. So this is a quite striking example, which is the example of the crocodile. So crocodiles don't have X and Y chromosomes to determine their sex, but the determination of whether they're male or female comes from one gene, which is modified. So the DNA of the gene is modified through epigenetic mechanism, and in particular methylation mechanism. And the epigenetic modifications are altered, are modulated depending on the temperature at which the animal is, so in the environment. So a crocodile can be male or female at different temperature, and this is not encoded by the DNA as if as it is for other animals or the human beings, but it is entirely controlled at the epigenetic level. Another example of how epigenetic is important for our body is it's understanding how different cells and different tissues are formed. So every single cell in our body harbors exactly the same sequence of the DNA, but starting from the same sequence, completely different cells can be formed. So a neuron depicted here, or a muscle cell that it's totally different shape and function. And the only difference that occurs between these two different cell types is their epigenetic adaptation of the DNA. If we go into the details of what happens at the level of DNA, and within the DNA, the building components are the genes, epigenetic is what cells use to turn genes on and off. So if the epigenetic structure of the DNA and the so-called chromatin is open, is loose, because the histones are not so severely tight, then genes can be expressed, can be turned on. However, if the chromatin, the DNA, is compacted and the histones are all wrapped together, then the genes will be turned off. So thinking about these and thinking how can we use these to target a brachyury in chordoma, we came up with the simple idea, which is, can we manipulate the epigenetics of chordoma cells to turn off the expression of brachyury, the presence of brachyury? So as I said before, brachyury is constantly on in chordoma cells, is there at high levels, and this is because there is an abnormal opening of the chromatin. If we could find epigenetic drugs, so chemical compounds that block this epigenetic modulation, and we could close off the chromatin, and therefore we could switch off brachyury, that would really be helpful in terms of treatment. So we knew this was possible. We knew this approach could be possible because there are a number of epigenetic drugs which have been developed in Oxford, which target a whole range of different enzymes in the so-called epigenome. These are probes, so chemical compounds, which have been developed at the University of Oxford in collaboration with this Structural Genomic Consortium, SGC, which is a public-private partnership based not only in Oxford, but also Toronto and Frankfurt. And the goal of this consortium is to create open access chemical probes, which are continuously updated. There are hundreds of these compounds, of these chemical probes, which are continuously updated and renovated, and they are offered as free open access to the whole community that would like to use them. One of the success story of the SGC compounds are the BRD4 inhibitors. And in particular JQ1. So these are epigenetic compounds that inhibit BRD molecules. These are BET promodomains. So these are enzymes that read those chemical modifications on the histones. And they've been shown to be extremely potent and very effective in reducing a variety of cancers, of solid cancers. And they've really gained a lot of interest in the last few years. And I think at the moment, most major pharmaceutical companies will have a BRD4 inhibitor, in at least phase 1 trial development. Many spin-out companies came out to develop further these drugs. We guess in the next 2 to 4 years, all the reports will be around definitely from all the clinical trials which are now being developed. So this is possible. So using epigenetic drugs to treat cancer is possible, but is it possible for chordoma? So we know this is the case. A few years ago, again in collaboration with Udo and Adam, we screened about 80 to 90 epigenetic drugs on chordoma cells. And these are the results of this first drug screening that we did. This is a heat map that shows the response of the chordoma cancer cell lines to the drugs. Each of these square is the results from one drug exposed to one cell line, and blue means no effect, whereas red means high killing efficiency. So first of all, you can see that many of these drugs were not able to kill the cells, so it's not an easy job. However, these group of drugs, the lysine demethylases, were really effective across a number of cell lines and were blocking the proliferation of the cells. And when we studied these drugs in details, it was very striking that the lysine demethylases were not only just killing the cells, but they were really shutting down, they were eliminating the presence of the expression of brachyury. So this really gave us confidence that this drug could be used to shoot an arrow on this Achilles heel for the disease. So this is what have been done before, but again, thanks to this funding and going forward, we wanted to explore even more compounds and to find new targets. So now Adam is going to give you an update on our latest results.
Hi, could you hear me? Should I actually take control over your computer then? Maybe try.
Yeah,
So,
you can approve.
did that? Oh.
Sorry, I think I need to.
If not, you can just continue to press next.
Yeah, maybe it's better.
Maybe it's better, and I'll just say next slide. So while Lucia is getting the slides up, so Lucia's mentioned that, you know, a few years ago, we had around about 80 different compounds, epigenetic chemical tools. And as she said, these are continually updated. And so now we have over 200 different epigenetic drugs that we can actually screen for. And this is one of the first exercises that we did as part of this new grant, was to screen these and then evaluate in essentially a test tube in an assay whether these drugs are able to kill chordoma cells. But in addition to the epigenetic libraries, we've actually started to curate lots of other different drugs for different chemical classes. So for kinases, PROTACs, drugs that modulate metabolism, as well as drugs that modulate something called ubiquitin proteostasis. And for instance, we have actually completed the kinase screen, and we are hopefully in the process of also evaluating the libraries as well. Next slide. Cool. So I think what I'm going to probably do is actually do a slight detour and maybe actually stop and give you just for a second a bit of background about why I wanted why I was motivated to become a scientist, why actually this project really excites me. So growing up, I was always fascinated and motivated to become a scientist because of the stories of, you know, like the likes of Marie Curie, Frederick Sanger, James Lovelock, who are really multidisciplinary scientists who've worked at the interface between biology, chemistry, and physics. And I've always I've always wanted to emulate this within my own research. And specifically, I think we can learn a lot more about whether a target will become disease relevant and tractable towards the clinic by combining our different skills together in different approaches within biology, i.e., you know, Lucia has talked about the chemical screens, and we leverage this with a number of the latest molecular diagnostic techniques, such as single cell, RNA sequencing, CyTOF. And you don't need to know what these things are, but what these techniques allow us to do is to generate lots of data surrounding the treatment of how the cells react to the treatment of the drugs that we're giving them. The problem is, it's really hard for individuals to make sense of all of this complex pattern of data. And so I actually spend a lot of my time writing software which integrates a lot of this different data types together to make sense. And so that's where the multidisciplinary aspect of this project comes in. It's taking all of this information from the chemistry side, the biology, and the physics from the actual machines. And then hopefully we can actually identify disease-relevant targets that we can say, actually, this is a good target, or actually almost more importantly, that this isn't a good target. We shouldn't waste our time pursuing this. Because I think a lot of the time in biology is spent on targets that never ultimately make it to the clinic. And I think if we use multiple different techniques, we can sort of be more confident in whether this is or isn't a target to take forward. So we don't waste money, essentially. Next slide. So our approach is to take these drugs and then treat them on different chordoma cell lines. But we don't just use one particular type of cell line. We actually try and use a number of them. And the reason for this is because, and this is inherent to all cancers, even within chordoma, these three different cell lines, which are MUG core, core, and UCH1, have been derived from different chordomas, ultimately from different tumors. And you would have thought that actually these cells should be behaving very similar. But the plot on the right-hand side is something called a principal component plot. And what this does is it measures the variability within the cells, just in their basal gene expression. If the cells were very similar and behaved similarly to each other, they would lie on the plot in the center. But actually, you can see quite clearly the cells are spaced apart. So this means that even though they are derived from the same type of chordoma, they actually do have inherent differences between the different cell lines. And actually, we can use this to our advantage because we can de-risk some of the, de-risk our strategy. So if we know that the compound is able to kill all 3 cancer cell lines, then we can be more confident that actually it's likely to work on all different chordomas. Next slide. So this is the results of the updated screen, and I'm not showing all the results. I'm only showing the drugs that we didn't have on the previous iteration of the drug screen. So on the left, you have the MUGs, the UCH1, and the core. So they're the 3 different cell lines, and we've treated it. So the bars going down are the different drugs, and the bars going right are 3-day, 6-day, 3-day, 6-day. And so we have treated all of these different drugs either 3 days or for 6 days. And then we've measured the ability of these drugs to kill the cells. And that's indicated by the purple. So if you see a line of purple, that means it's killed all the cells. That drug has killed all the cells at each different day. And we've identified several new targets that weren't previously on our screen that we did a few years ago. Next slide. So I'm just going to go briefly through a number of these compounds that we've identified. I'm not going to go through all of them, but specifically, one of our hits was BRD9. And this is a component of something called the SWI/SNF chromatin remodeler. And you don't need to understand what that actually means. Ultimately, what this chromatin remodeler does is it changes the 3D conformation of the way your DNA is positioned within the nucleus. And if you change the 3D conformation, you can actually ultimately change the gene expression. Specifically, as Lucia said previously, the Brachyury gene. This drug changes the 3D conformation and then results in a reduction in Brachyury. And this is the studies which we're actually following up mechanistically. Next slide. So as I said, you know that the compounds are continually updated, and actually even from when we did the initial screen, which actually wasn't that long ago, a few months ago, there are new compounds that are even more specific and selective for bromodomain 9. Which we are now following up, as I said, in more mechanistic studies. Next slide. Another one of the screening results was actually halofuginone, which we actually did identify a number of years beforehand on the initial plate, the MAS1392 and MAS1805. And interestingly, these aren't actually epigenetic compounds. These interfere with something called tRNA synthetase. It's not terribly, so the reason why they were put onto the plate, the epigenetic plate, was to study something that we were seeing within the lysine demethylases, but it just turned out that actually they seemed to kill the chordoma cells very effectively. What these drugs are, they're derivatives of a Chinese herb, Brachyury. And this Chinese herb has been used in traditional medicine as an anti-malarial for many, many generations. And the contribution of the new screen is that we've actually able to identify 2 new derivatives of the MAS compounds, which are actually even more potent and more selective, and so potentially more tractable for drug development. One thing to note is actually with these compounds, we are following this up in vivo with the Chordoma Foundation as well. Next slide. So as part of this grant, what we've been doing is the first stage is to find out how potent and how selective these compounds are. So that is, we do this by taking a very high concentration of the drug and diluting it down, and then treating the cells with the different dilutions. And you can see you'll get, well, actually I haven't got a pointer, but you'll see that you get a dose response curve. And you can work out what we call the inhibitory 50% concentration. And then we use that at all subsequent experiments, because you don't want all the cells dying, because you would want to actually look at the cells going into death, not at the end stage. So if you move to the next slide, Lucia, please. So the next thing that we've done is we've actually looked at the effect on Brachyury. Just to remind you, this is the Achilles heel of Chordoma. If we're seeing a reduction in Brachyury, this is likely how the cells are going into apoptosis or are being killed. And this is exactly what we do see. Lucia is highlighting that when you treat with Halofuginone, you get a reduction versus DMSO. So mRNA is essentially the messenger that goes from the nucleus, from the DNA, to the cytoplasm, and then it ultimately instructs the cell to make protein. So do we see a reduction in protein? And actually that's exactly what we do see. So these are Western blots, and these are a particular technique for looking at protein expression in a cell. And when you compare DMSO to Halofuginone, you can see a clear reduction in the expression of Brachyury. Next slide. But we aren't just interested in looking at Halofuginone. We actually want to work out the molecular mechanism, because if you're ever going to take a drug forward to the clinic, you need to know what its mechanism is like, or what the mechanism of how it actually kills is like. So we employ a number of different techniques in our arsenal. One of them is using RNA sequencing. And so we can measure the gene expression in the cells that are treated with the compounds simultaneously. So we could capture the whole gene expression at the same time. And one thing that we've noticed is that you see a significant stress response that we think is driving ultimately the cell death and the Brachyury reduction. And this is something that we're currently following up at the moment. In addition to this, we're also applying, as I mentioned before, a lot more different molecular techniques to try and build up a picture of what actually is going on inside the cell when we add the compounds onto the cells. And what we want to get to a stage is we want to get lots of data surrounding this topic so we can then mine it, ultimately to say, is this a good idea to take forward, or is this a bad idea? Because as I mentioned before, I think identifying a bad target is probably more important, if not, well, equally important, if not slightly more important than identifying a good target, because lots of research and lots of money is invested into ultimately the wrong target. And you really want to avoid that. So I think in the pharma industry, they have the saying of, you fail early and fail spectacularly. I suppose that's what we're trying to do. If this is not going to work out, we want to fail now rather than, in a few years down the line, because the drug discovery stage takes, up to 10 years. It's quite a lengthy period of time, and that requires every single, day, it requires more money. Next slide. So I just want to mention one of the techniques that we're going to be applying to this project, and specifically, this is something called single cell measurements of the RNA. So normally, as I showed before in one of the previous slides, looking and capturing the gene expression, what we normally do is we take a bunch of cells from a tissue, or we take some cells that we've cultured in the presence of a drug and the absence of a drug, and then we mash those up together, we extract the RNA, and then we compare one pool of cells to another pool of cells. This is akin to essentially blending a smoothie up with lots of different fruit and then giving it to your friend to work out what's in that fruit, and then comparing two different smoothies. It's really difficult to do. You can maybe identify, well, it's got strawberries in it, it's got banana, but you may miss, you know, the 1 or 2 blueberries that are in that smoothie. What we want to do is to apply techniques which we can go through that smoothie and then identify every single constituent. And that's the same as what we want to do on the cells. We want to measure the gene expression in every single cell, because that's ultimately where epigenetics works. It doesn't act on so Lucia mentioned before that, you know, your muscle cells are very different to your eyes. Why is that? Well, that's because of epigenetics acts at the single cell level to make these tissues very different. Therefore, we need to study this, the drug's effect at the single cell level. And so, next slide. So to do this, we actually use droplet-based sequencing approaches. So these are microfluidic devices, which you have cells and oil, and then a lysis buffer moving in, and cells are then encapsulated within droplets, and then all the chemistry takes place within this droplet. So you capture the RNA, and then you can then sequence it. Next slide. And so we've actually been doing this, well, at least in my group, quite successfully over the past 1 to 2 years. So this is actually on an inflammatory project where we took the drug GSKJ4, which is an epigenetic drug, and we stuck it on what we would have thought are quite homogeneous cells. So these cells, you know, should behave very similarly. But actually, when you add the drug, you see very different effects. And this is, without going into too much detail, you can visualize this on this dot plot on the right-hand side. So these different colors of dots are essentially the different populations that you get coming out and the gene expression changes that occur when you add the drugs to the cells. And what it's saying is that the cells are behaving very, very differently depending upon, well, they're not all behaving the same way. And I think this really blends into the fact that we really need to start studying cells in drug, these epigenetic drugs at the single cell level. And this is a technique that we will be applying to the chordoma cells. Next slide. So I think the conclusions from the screening so far, we've actually identified 2 novel halofuginone derivative compounds that we're currently following up both within the Bone Cancer Research Trust grant on the in vitro side, but Udo Opperman and Adrian have a grant to follow this up in vivo with the Chordoma Foundation. We've identified BRD9 inhibitors as targets that we're currently following up as part of this project. Specifically, what is the mechanism of how this compound or these compounds are killing chordoma cells? We actually have identified several other targets, but unfortunately, we just don't have the scope to follow these up within this grant. It would be great too, but what we're going to do is then apply for further funding to facilitate this. In addition, I haven't even gone into any of the data, but we've also completed the kinase screens. We've identified 10 novel new kinase targets, which would be nice to follow up. But again, given the short length of the grant, we don't think we'll have scope in this to follow up. However, I think it's really good food for thought that there are potential novel targets that we could take forward going into the future. So if we go to the last slide. And so I suppose this is the elephant in the room, and this is the impact on coronavirus. So I think I wanted to address this because I think there's actually some hope in this, because like everyone else, I think everyone has been affected by coronavirus. Everyone in the world has been in lockdown, including scientists. So our labs have been shut for non-COVID related research for the past 3 months. It could potentially have been devastating. I've personally been involved in COVID related research in Oxford. And so my time hasn't been able to be spent on this, and Lucia also. However, we have mitigation strategies, so this shouldn't hopefully have so much of an impact on our research. Specifically, the research assistant who is doing quite a lot of the work for this grant has been put on furlough. And also the cost extensions that we're getting from the Bone Cancer Research Trust are going to be massively helpful to help us finish a lot of the work that we proposed in this grant. So hopefully that gives you some hope that we hopefully shouldn't be affected by this too much. So I'd just like to thank, obviously, Lucia, who I'd love collaborating with, Adrian and Udo, as well as a number of different people within the group, particularly Graham Wells and Alexandra and Martin on the Oxford side. And I'm sure Lucia wants to also comment on the UCL side. And also from me, the Bone Cancer Research Trust, everyone who has donated to the charity, and also the other funders who funded parts of the work as well, particularly the Chordoma Foundation. And so, Lucia, maybe you want to.
Yeah, no, definitely all the members in the group, and also the clinicians, and actually also all the patients that donated their samples, because without that, this research and none of the research that we're doing would have been possible. So I think, Adam, what do you think? I think
that's it, yeah.
And
if there are any questions, that would be great.
So, Lucia, do you want to just stop your screen share?
Yeah.
There we go. Thank you. Thank you ever so much, both. That was really, really great and really inspirational. It's really, really inspiring to see, the promising results that you're starting to see, and it's great to know that there's other scope for the work to continue as well, should the funding be available. So it's also really, really, as a charity at BCRT, we're so proud of being collaborative and working with other organisations. And I think this project's an excellent example of where we're funding one part, the Chordoma Foundation are funding another part, and how multiple grants help support one another, and other work as well, like Chordoma UK, they're funding. It's really fantastic to see all that coming together. So I'm going to hand over now to Vicky, who has she has all the questions that were asked in advance, so she's got those to put to you, and also take questions that have been asked in the chat function. If anybody else does have any questions, please do pop them in the chat now as Vicky starts putting the questions forward. Over to you, Vic.
Okay. Thanks. Thank you. It was really interesting. As usual, we have the questions that people have sent us in advance. So I'm going to put those to you first, and then I'll go through the ones that I've been looking at the chat throughout your presentation. So I'll start with a question that we received from Lorraine Pike. And she's asked, are there any advances in knowledge in the UK or anywhere else in terms of being able to identify recurrences of chordoma and therefore being able to adapt treatment?
Lucia, you're muted.
I'm here now. So of course, disclaimer is I'm a basic scientist, so I can answer more on the scientific point of view. But I think there are continuous changes and advancement that can be done and that we can do. In terms of the identification, I guess the imaging is particularly important, and any decision that will be made is not just made by one person, but is a multidisciplinary team of oncologists and radiologists and different experts that within the sarcoma specialized centers will make these decisions. There are some research that are being followed up at the moment. One of the promising areas, it's the identification of tumor circulating DNA. We're not there yet for chordoma, but there are a number of things that will be done. But at the moment, really, it's down to the interaction of the multidisciplinary team and doctors within the sarcoma centers that can really judge also on the specific of each single patient. I hope that's been useful.
Thank you. Thanks, Lucia. The second question that we received in advance is from Helen Gordon-Smith, and she's asking whether there is or do you know further research into familial incidence of chordoma?
Yeah, so I think it's you know, as I mentioned, there are about 40 new chordoma cases every year. Familial cases are just a handful. There have been studies that have really tried to look into the details of the genetics and the genetic predisposition to the development of chordoma. They've been really important because I haven't mentioned in the introduction, but those families carry amplification, so duplication of the Brachyury gene, which seems to be a predisposing condition to develop chordoma. And knowing this from family has prompted us to look for the same type of genetic alterations in sporadic cases. And only by looking deeply, you know, looking at the whole genomes and looking with very specific techniques, were we able to identify very similar changes that we wouldn't have known unless we knew the data from the familial cases. I think at the moment lately there haven't been any new discovery. Things are made, and I think now also with the there's going to be quite a lot of attention also to the familial predisposition, looking at the sporadic cases as well. And I'm sure you're aware of the 100,000 Genome Project, where both the tumor DNA and the germline of patients will be sequenced. I think there is quite a lot of hope, you know, and there is definitely scope and energy to look for predispositions also in these cases. Again, I hope I've answered your question.
Thanks, Lucia. I was going to follow that up with something that related. So the genomic profiling of chordoma tumors for patients, is that going to be a standard practice, or do you know when would that be a standard practice in the UK for patients?
Maybe Adam, you can confirm, but my understanding is it is standard practice as one of the sarcomas. So it should be followed up within the 100,000 Genome, within GEL project.
One of the things that actually, as a cancer, it makes us as a scientist, our job at understanding the disease mechanism or the cancer mechanism slightly easier, because ultimately it's driven by the activation of Brachyury. And there are very few cases that I know of, but I'm not the chordoma expert, so this is just my own reading, that ultimately if you have expression of Brachyury, you have chordoma. And so that in effect makes our life slightly easier, because there's not sporadic mutations that are occurring constantly within the tumor. So if you can inhibit Brachyury, you should inhibit the tumor. It's not like, you know, metastatic breast cancer or you know, significant multiple myeloma, for instance, where you get huge turnovers of cells, and each cell will be clonally different from the previous one beforehand. That doesn't seem to be an issue within chordoma. And so that's my comment on that, if that makes sense. Lucia?
Yeah.
Yeah, I'm going to ask you a couple of questions, which is sort of background knowledge to make sure everyone's on the same page for everybody. So could you explain, just for everybody, what the cell line is, and also what the homogeneous cells are? Just very briefly.
Yeah.
Just, Lucia, do you want to go for that? Or
Oh, yes, of course. So we give some of the terminology for granted. So when we talk about a cell line, we talk about some cells that were taken from a tumor sample and were dissociated. So they were, like, the tissue was minced and they were spread on the plastic devices in the lab. And we talk about a cell line when these cells start growing indefinitely, so they can propagate, they can divide and grow indefinitely in the lab. So we are working here, you know, it's cancer scientists, we work on cell lines that were established 30 years ago, and because they're lines, they can still grow in theory forever. And this allows many different labs around the world to work on the very same material and therefore make perfect comparisons of experiments and the results to be repeated over and over again. So this is the advantage of having cell lines, so that can be propagated forever. What was the other question? Sorry, I forgot.
No, in a way it follows on to a comment that Liz Brownhill, or a question that she placed, whether, so she was wondering whether the results that you see on immortalized cell lines, have you tried to look if you get similar responses in patient-derived cells?
Yeah, so I mean, so the initial idea of having a cancer cell was, okay, we have one patient, one cell, one cancer cell, we use it, you know, we propagate it, we use it forever, we'll get the answer for all the rest of the patients that have the same type of tumor. But the more we've been doing our research, the more we have realized how every single patient is different. And even within the same tumor, every single cell has a level of difference, and therefore it's so complex. So that, as Adam has explained, we're really keen on all our drug screening to use 3, 4, 5 at least different cell lines to make sure we catch what what will more likely hit across more samples and therefore hopefully more patients, and we also want to go into the details, not only look at the surface of what's going on, but really look into the details. And the more we dig into the details, the more we will understand how these drugs work, and hopefully more efficacious responses. Adam?
If I could just suffix that as well, by the reason why we use cell lines goes back to the reason. So patient samples are incredibly precious. You don't want to waste them. So the reason why we use cell lines is because we're screening many, many different compounds. And so we take these immortalized cells as essentially an assay or a model system where we can study the chordoma very effectively and very repetitively. And it doesn't matter if, this experiment, we somehow, I don't know, if we put the wrong media in, for instance, or we can scale the experiment up and we can use, 400 different drugs rather than screening 200. And it's to avoid that, unnecessary waste of patient material, which you want to use the patient material when you've really worked out that, okay, this drug is good. This is the mechanism. Does it then work in primary human samples? And that's when you would use that particular type of tissue. And we've just finished the screens. They've gone quite well. We're going into mechanistic detail, and then we will, if it pans out, move into primary tissue.
Yeah, makes sense. So I'm going to follow up with another question, which, so you've tried with your cell lines, which are Chordoma cell lines, and you get a really nice result with some of the compounds. Have you used as a comparison other cancer cell lines that don't have Brachyury, and do you have a differential response?
Adam, do you want to?
Yeah, so I've been working in the chemical biology space now for about 4 or 5 years, and we've screened many different cancers, from pancreatic to multiple myeloma, bone cancers, and then inflammatory disease. So rheumatoid arthritis is what I previously worked on, and it really is cell type dependent. You get some hits which are really strong in certain cancers and not in others. And this ultimately depends, it's driven by the underlying gene expression response that you get in the different cells. So yes, you see very different responses depending upon the cells that you add in your screen.
Yes, I was thinking of, whether it was specifically linked to the presence or not of Brachyury, the compounds at screen.
So we know that some of these epigenetic compounds will be directly inhibiting Brachyury for definite. So some of the chromatin remodelers, and particularly, that's how we think maybe BRD9 might be working. But for some of the other compounds, we think it's probably an indirect mechanism, particularly the halofuginone, for instance. We don't think it's being driven directly by the reduction in Brachyury. We think the stress response is actually driving the brachyury reduction.
Yeah. Yes.
Yeah.
Makes sense. Yeah. Thank you. I have a question which I'm going to read straight from the, because it's quite a lot, from Professor Sue Birchall, and she's asking about BET proteins, BET proteins, yeah. And she's saying BET proteins interact with acetylated histones in active regulatory domains, whether they're promoters and enhancers, and promote Pol2 activity to regulate gene expressions in all cells. How do inhibitors of BET have cell-specific effects? That's the question. Or in another way, what is the basis for the therapeutic window that means inhibitors of BET are attractive treatments?
So maybe I can answer that one?
Yeah, sure.
Okay. So actually, it's a fantastic question. It's phrased really nicely as well. So when the BET bromodomain program started in the SGC, no one really was interested in it because everyone thought, particularly pharma industry, why would you ever want to inhibit a BET protein? Because it's so, the cells need it. Surely it should be really, really important for every cell within the human body. And how do you get selectivity? By targeting something that is ubiquitous within every single living cell. But I think when the numbers of years that we've been using with these bromodomains, they've now actually gone into clinical trials, and they've been found to be actually relatively, they get their selectivity because each different cell has an underlying different gene expression, different enhancers. And so when you use these compounds are more effective in highly proliferative cells. And so it gets selectivity in certain cancers where they're really proliferative. And so these BET bromodomains are active at a higher rate than they would be in your normal tissue. And to an extent, this is how normal chemotherapy works as well. It's actually targeting the highly proliferative cells, and specifically the cells where BET is constantly required for the turnover of these cells. Yeah. So that's, in effect, it's not the only mechanism, but it's one of the explanations of how you can get selectivity within cancers versus non-cancerous cells. And I hope I've answered the question as best I can.
Thanks. Yes. So it's a connected question in a way, because it relates to the epigenetic modulator. So I have a question from Amin, and he's asking about how these epigenetic modulators would affect other cellular functions, and therefore how specific the treatment would be.
So for instance, there is a slight, I think we do know the mechanism of chordoma. It's brachyury related. The problem with chordoma is that it's a slow growing cancer. And that's the reason why chemotherapies just really don't work very effectively. However, what we can see is that even at really sub-micromolar doses of some of these compounds, specifically, the mouse compounds, and the degrader of the BRD9, we can get the concentration right down, and it is specifically having an effect on the brachyury. And hopefully, the idea is that if we use it at a really low dose, it shouldn't affect any of the normal cellular function within the body. That's the theory anyway. In practice, the drug development process is fraught with many, many different obstacles, and it may just not work out. And it may not work out for many reasons that you wouldn't think of. And at the moment, we just don't have that information. We have to pursue a target. And I think it goes back to the problem of trying to fail early. If we can try and find all of these problems with using these compounds, not just in chordoma, but in other cells. So that's why I think the other work we're doing in the group on other, inflammatory cells, on epithelial cells, we're learning from these different cellular systems, which hopefully can then inform the work that we're doing in chordoma. If you understand, if I'm being clear on that. But.
You are, yeah. I have another question with, so you mentioned different treatments for chordoma. So one of the options is targeted therapies, and you mentioned kinases. So I wonder if you could, I know it's, I'm asking you a huge topic to condense into a few words, but could you briefly explain what kinase inhibitors are?
Maybe Lucia wants to mention that.
Thank you, Adam.
I can,
it's an unfair question.
I can go for it essentially if you want, but I'm just quite worried that my definition of kinases is going to be probably a bit too low level and too much detail, but I can mention what kinases are if you want. So kinases are essentially, they're enzymes which catalyze the transfer of phosphates. They're essentially energy transmitting molecules. They initiate phosphorylation cascades in cells, and they act quite high upstream within the cellular structure, and they initiate signaling cascades, which then ultimately, each different signaling cascade has a different effect, but they ultimately, a lot of them end up changing gene expression. And the way they do this is through enzymatic catalytic activity acting on certain different proteins within cells.
If I can, so if I could explain, it's that though the molecules which are, their role in the cell is to basically transfer the signal, the information from the outside of the cell. So, hormones and all other sort of signal, how the cell perceives the presence of these external signal is through kinases. So they are these transmitters of the cell. And as Adam explained, is through these phosphate and energy transfer mechanism. And they are, because they're involved in so many different responses to so many different stimuli and signals, are one of the most, favorite targets for many of the pharmaceutical companies and all the drugs that have been developed so far, because they're involved in a variety of different cellular responses.
Also on the chemistry / structural biology side, they're also really a lot easier to develop small molecules against. So that's some of the most successful pharma programs have been on kinases, because you can very, very effectively target small molecules to inhibit the enzymatic catalytic activity.
So a few years ago, within the group of Adrian Flanagan, we did a kinase screen, similarly to our epigenetic screen. We used, I think it was 1,300 kinase inhibitors. So again, because there are a variety, so the companies have been developed kinase inhibitors over the years, a lot more than epigenetic compounds. And we screened 1,300. And it was very difficult to find something that was really effective in chordoma cells. So the really only class of drugs that we found was the EGFR inhibitor that then we followed up and studied in more details. And now there is a clinical trial going with this, with the EGFR inhibitors.
Interestingly, I passed our screens to Udo for the kinase screens, and he got excited by 2 of them. So specifically 2. I think we had about 10, maybe more hits from the last kinase screens. So we'll see. So Udo's a biochemist by training. So when he gets excited about things, you usually take notice. So we'll see what he says.
I have another question regarding kinases. So you mentioned EGFR and some PDGFR like Gleevec and imatinib, which is very well-known drug that people actually is used for. So these are very selective. So they try to target only just one of those enzymes that you mentioned. But they sort of have those, they have effect, but not a complete effect. So do you think it's more appropriate to go for a non-selective kinase inhibitor or a combination of different ones? Do you have a view on that?
I don't think you'll ever get specific kinases, so that's just a comment from. So we've actually the problem with the kinases is that actually they share quite structural homology between each other. So you target one, you inadvertently potentially target others as well. So you have to be very careful. And even the drugs on market, even though they may say they inhibit one particular kinase, I bet you any money they're inhibiting maybe 2 or 3 or maybe more.
Yeah.
So that's the comment from my side. I don't know, Lucia, do you have something to say on that?
Yeah, I don't have any other comment.
Yeah. There is another question which is completely unrelated to this, but it relates to radiotherapy. So it's a question from Louise, and she wanted you to comment, if you could, on the future of, or the effect that proton beam therapy would have on the treatment for chordoma patients.
Again, it's you have to take this answer from myself, not as a clinician, but as a scientist. I think the community, the scientific community, is really putting a lot of hope, and I think in a kind of supportive and reasonable way into looking into proton beam therapy over photons. Now, well, just behind the corner here, UCLH is building this huge proton beam center, and I think it's very important for us scientists as well, because there will be the possibility to access the center and do some research. So we will be able to do some in vitro studies, also for chordomas, using chordoma cell lines or cells to look at what the effect and to look into the mechanisms of action of the proton beam therapy. I think generally, it's something that we will start studying, and we will hopefully see an advancement, at least a new opportunity also for chordoma.
Thank you. I have one very last question, which relates to, again, us treating brachyury indirectly or targeting brachyury indirectly, which refers to immunotherapy options, like, for example, cancer vaccines. Would you be able to comment a little bit on that? Because some people might be interested to hear about it.
Yeah, so this is, so of course, the research in brachyury, so chordoma in a way has been a pioneer because the research that has stemmed from from looking at brachyury in chordoma has actually opened the way for another route of research and looking at the expression of brachyury in other tumors. So as I said, really chordoma is 100% of chordomas will express high levels of brachyury, but there are some scientists, especially in the United States, where they have identified that other cancers, and in particular specific subtypes of cancer, very aggressive ones, will also start expressing brachyury, and therefore there has been a scope for looking at the role of brachyury also in these cases, and using brachyury to using brachyury to hit the response of the tumor to the immunological response to the tumor as well. So there are groups that we've not directly been involved with the studies, but there are groups who are developing a vaccine where you can take basically brachyury, mince it, and inject it to invigorate a response, an immunological response against the tumor. I think the clinical trials are running at the moment, so some research and some progress has been made, and I think that the answers will come up in the next few years.
Thank you.
Is that it, Vicky? Have we gone through all the questions?
I think I managed to get through all of them, yes.
Oh, well done. Okay, so we've run over a little bit, everyone, but we hope that was really valuable and insightful for everyone on the webinar. Thank you, first of all, to Lucia and Adam. We really do appreciate you taking the time out to join us. We know that you're both very, very busy, but we really do appreciate it. And thank you, everyone that has joined us from your living rooms or your offices or wherever you are in your kitchens.
Well, thank you for the invite, so thank you.
Thank you for inviting us. It's very important for us.
You're very welcome.
Thank you. Thanks a lot.
once again, I think it is really important for us as a charity just to acknowledge that, you know, it's great that our work combined with the efforts of, you know, the Chordoma Foundation, Chordoma UK, Sarcoma UK, all coming together to really, really move research forward for chordoma patients. Somebody made a comment that it really is, it really does give hope to current patients and families, and I think, you know, Lucia and Adam, you should really take that to heart. So well done, and thank you. It's great to have you working on this research for us. So everyone, this was our penultimate webinar. Our last one is next week, and we're going to be joined by Dr. Elizabeth Roundhill, and she's going to be talking about progress in research mainly around Ewing's sarcoma and bone cancer stem-like cells. So please do join us for that. We look forward to welcoming Dr. Roundhill next week. But thank you all once again, and we will be sharing the slides and the presentation via YouTube probably on Monday or Tuesday of next week. So if you want to watch it back, you'll have the opportunity. But thank you all. Have a lovely weekend. Take care.
Thank you.
Bye.
Bye, everyone.